All right, so we are in chapter 8.6, and we are talking about photosynthesis. So photosynthesis, I'm sure that kind of you're familiar with photosynthesis, having taken prerequisites prior to this class. But remember that photosynthesis is this process where we are taking the sun, our energy from the sun rather, and converting that into chemical energy. So in the terms that we have been using in this class so far, this is where we have a biochemical process by which phototrophic organisms convert solar energy into chemical energy. So remember, we did speak about different terms. When we're talking about things like heterotrophic, remember hetero meaning different and then trophic meaning a source of nourishment. Remember that we have heterotrophic organisms like ourselves that rely on chemical energy that's found from other organisms, or that we get from other organisms, complex organic compounds. And so these complex organic compounds come from autotrophs, meaning they make this carbon with themselves, and then also photoautotrophs is what we're talking about here, are phototrophic organisms that are going to be taking this sunlight, converting this solar energy into chemical energy. So remember, these are phototrophic. So our phototrophic organisms. So remember, when we talk about phototrophic organisms, we're talking about things like plants, but then also like cyanobacteria and other photosynthetic bacteria and microorganisms. So when we take a look at photosynthesis, it comes in two stages. It has two stages to it. The first stage is called the light-dependent reactions. And then the light-dependent reactions, as its name implies, this is when we use light, and light is absorbed by different pigments in the membranes, in the photosynthetic membranes, and then it's converted into this stored chemical energy, so this short-term stored chemical energy. So light is absorbed by pigments in membranes and converted into stored chemical energy. So then this stored chemical energy is going to be used in the second stage, which is the light-independent reactions. So light-independent reactions. And so then in this, we're taking this chemical energy and then it's used to assemble sugar molecules using carbon dioxide. So it still relies on having light. So it says that it's light independent because it's not necessary to go through these specific steps. But of course, it relies on there being light because we have to get the chemical energy from the first step, the light independent or the light dependent reactions, to get these short-term chemical energy molecules like ATP and ADPH, and utilize those then in the light-independent reactions. So in this case, we're saying the chemical energy that we just got here is used to assemble sugar molecules using carbon dioxide. All right, so then these reactions are used to drive energetically favorable process. So these reactions here are used to then drive this process called fixing carbon dioxide into this organic sugar. And we're going to take a closer look at how we do that. Before we take a closer look at how we do that, though, let's take a look at where this is occurring. So these different structures. So our photosynthetic structures. So first, if we take a look at eukaryotic organisms, The first one that we can talk about is probably the most familiar, which is the chloroplast. So this is an organelle in eukaryotic organisms, which is originally from an endosymbiosis. So from a photosynthetic bacteria that probably somehow merged with a different cell, then becoming our photosynthetic eukaryotic cell. So the chloroplast itself then would be the organism that it was previously. So this photosynthetic bacterium that now is considered an organelle inside of the eukaryotic cell. So an organelle in eukaryotic originally from endosymbiosis of a photosynthetic bacterium. So the chloroplast, we'll take a look at the structure in just a moment. Within the chloroplast we have thylakoids, and the thylakoids are these disc-shaped photosynthetic structures within the chloroplast. So within chloroplast, they're disc-shaped photosynthetic structures. So I guess we can kind of draw it here. So we have our chloroplast, which we believe came from endosymbiosis from a photosynthetic bacterium. Inside of this chloroplast, we have thylakoids, so disc-shaped little discs where we have photosynthesis happening. So we have these little tiny discs in there. I drew them in this way because what we have are called granum, or agranum, or grana, plural. I can put that in there, grana. And these are stacks of thylakoids. So this right here is the chloroplast. This right here is a granum, singular. And then this individual disc here is the thylakoid. And then we have, lastly, the stroma. And the stroma is just the space that's surrounding the granum within the chloroplast. Within the chloroplast. So then this space out here is the stroma. And this is important because when we talk about photosynthesis and we're talking about the two steps, the two stages, we're going to see that some of that is going to take place within the thylakoid in this stack of granum, and then some of this is going to take place in the stroma, so the space outside of that. And of course in my drawing this is overly simplified. We have lots and lots of grana in here, lots of stacks of thylakoids. So lots and lots and all kind of crammed in there. So this is when we're talking about eukaryotic organisms. So we can think of plants in this case, for example. Now, if we take a look at prokaryotic organisms, they have photosynthetic membranes similar to thylakoids, but recall that prokaryotic organisms do not have organelles. And since they do not have organelles, this is just an infolded region of the plasma membrane. So we don't have a separate section. We don't have a separate organelle. Instead, what we have are photosynthetic membranes. Membranes are infolded regions of the plasma membrane. And so in some instances, these actually are called thylakoids. In others, they're not. So, for example, called thylakoids in cyanobacteria. So within this infolding of the plasma membrane, what we have are photosynthetic pigments. These are molecules that are embedded in the membrane, and then they're organized into what are called photosystems. So we have photosynthetic pigments that are embedded in the membrane and organized into photosystems. And so we have these photosystems, which we're going to look at in much more detail. But some examples of our photosynthetic pigments are, we have bacteria chlorophylls. And these are green, purple, and red colors. In these different colors, we have our carotenoids, which give orange and red color. We have chlorophylls, which we see a lot in our plants and eukaryotic photosynthetic organisms. Our phycocyanins, which give a blue color, and then our phycoerythrins, which give a red color. Erythrocyte, erythrins, red blood cells, red. So we have all of these different photosynthetic pigments. And perhaps you can relate these to the changing of the colors in the trees, because what's happening is as the energy is kind of being pulled out or drained out of the leaves so that they can fall in fall or autumn, sometimes some of these pigments are left behind in the leaves. And it depends on what pigments the tree is going to bring into its trunk. It depends on what pigments are left behind, but this is why we see some of the things like the carotenoids aren't often brought in, but the chlorophylls are. So chlorophyll is typically a molecule that the tree brings into the trunk to store up for later. And then what we see are the carotenoids left in the tree later, and that's why we see the orange and red of the leaves as they fall in the autumn. However, when we talk about microorganisms, we can talk about actually seeing these bacteria being in these different colors. So we have bacteria that are in big pools that turn the pool or the water looks very purple, for example, with the bacteria chlorophylls, or can look orangish red, depending on the carotenoids or the phycoerythrins, or bluish because of the phycocyanins. So we have these different photosynthetic pigments. And these photosynthetic pigments, again, are embedded in this plasma membrane. This plasma membrane has enfolded to the interior of the actual organism. And then we have different parts to this photosystem, which is the last piece here. So the photosystem has a light harvesting complex. And this light harvesting complex is comprised of multiple different proteins and the pigments, and then these are what absorb the light. And then they are excited by the light, so the energy that it takes in because of the light excites the molecules, excites the electrons, and energy is going to be transferred to the reaction center. So not the electrons here, but just the energy of those excited electrons is going to be transferred from one section of the photosystem to another section until it then gets to the reaction center. So our light harvesting complex, we'll say multiple proteins and pigments that absorb light and are excited, and the energy is transferred to the reaction center. So then the second part here is the reaction center. So the reaction center is what contains a pigment that can oxidize as it's excited. So it loses an electron, and that electron is going to move to another location, and we'll talk about that in just a moment. So the reaction center actually contains a pigment that can oxidize as it is excited. So then it losing, well, maybe not one, let's just say losing an electron. Losing an electron. So we are in the light harvesting complex, these multiple proteins and pigments are absorbing light. They get excited, so these electrons are moving around very fast and moving to multiple different levels. They're excited, and then that energy is all going to be kind of bouncing around and transferred to the reaction center. In the reaction center, then we have this pigment that can oxidize as it's excited, so all this energy gets to the reaction center, and it loses an electron, and it can move that electron to the next step in the process. The next step in the process is the electron transport system. So the electron to the ETS. So this ETS, it's a different type of ETS. This ETS is similar to cellular respiration, and it's embedded within the photosynthetic membrane. But what this does is it can move the electron throughout this electron transport system, the ETS, and it creates this electrochemical gradient. So just like our oxidative phosphorylation that we see in aerobic respiration, the ETS of the aerobic respiration, we see this electrochemical gradient being created, and then this allows ATP production. Now, we actually have two different photosystems that we're going to talk about, and so there are different ETSs associated with the different photosystems, and we'll talk about that in just a moment. So the electrons are transferred to the ETS, which is similar to aerobic respiration, or rather just respiration, doesn't even have to be aerobic. So similar to respiration, and is embedded within the photosynthetic membrane. It creates an electrochemical gradient that allows ATP production by chemiosmosis. And this part, this ATP production by chemiosmosis, is going to be photophosphorylation, which is a type of oxidative phosphorylation. And we're going to talk about that in more detail. So we see this photosynthesis in eukaryotes that occurs in the chloroplast with these different light harvesting complexes with the reaction center, and then we have these ETSs. And we also see these in prokaryotic organisms, but we see it within these infolding regions. So now that this electron has been removed from the reaction center and moved into the electron transport system, we now need to get a new electron in that reaction center. And this is where we come upon two different types of photosynthesis. So we have oxygenic photosynthesis and anoxygenic photosynthesis. So when we're talking about oxygenic photosynthesis, then, as the name implies, oxygen, right? This is when water is going to be split to supply that electron to that reaction center. So it's going to split water. That oxygen is going to be in that reaction center, or is going to supply the electron, not the water itself, or the oxygen itself, but the oxygen is going to supply that electron. And then we have O2 as a byproduct, which of course is oxygen gas. So if you think about this in terms of a plant and the photosynthesis you're familiar with, plants go through oxygenic photosynthesis. So this is when water, and we know that plants need water, for example. So water is split to supply the electron to the reaction center as a molecule, and that is termed H2A. And the reason that it is H2A is when we talk about anoxygenic photosynthesis, that's because the A in this case won't be oxygen, as it is here in oxygenic photosynthesis. But so then our oxygen, our O2, is a byproduct. So it loses an electron, basically it gives up that electron to the reaction center. When it does this, then we have O2, or oxygen, as a byproduct. So that's why we kind of already know that we have to water plants. Water these plants. The plants take up the water. That's an important aspect of having a plant. And then the sun out there has to be on the plant. The plant has to get sun. It needs to get sun for the first piece that we've already talked about, the light-dependent reactions that we'll talk about in more detail. But this is when we have that energy coming in from the sun that's increasing the excitability of the electrons, increasing the energy of the electrons, that then goes to that electron in the reaction center. So then this is where we get the water part from, is that then that water is split, the electron comes from the water, and then the oxygen is a byproduct, and we know that plants release oxygen, and then later we're going to talk about how the carbon dioxide that we know plants need come into consideration here, or come into the process. So again, I'm talking about this in terms of plants because I think that most of us are most familiar with plants. But then this actually specific process, oxygenic, occurs in plants and cyanobacteria. So I'll write that down, plants and cyanobacteria. So then we also have, however, anoxygenic. So anoxygenic then is going to be an meaning without, and then oxygenic, so without oxygen. So this is when other reduced compounds are going to supply the electron to that reaction center. So in this case, we do not have oxygen as a byproduct. We have some other byproducts, so something like sulfur or sulfate byproducts. And this is because oftentimes we see hydrogen sulfide or thiosulfate as the electron donors, and then we get our sulfur or sulfate as the byproducts. So in this case, similar to what we've seen previously, this just means when something other than oxygen or something other than water. So when other reduced compounds supply electrons to the reaction center. So oxygen is not a byproduct. So energy is not a byproduct. So for example, like I mentioned, we can talk about hydrogen sulfide or thiosulfate. This is thiosulfate. So thiosulfate, so these would donate the electron, and then what we would end up with as byproducts in this case, we would end up with elemental sulfur or our sulfate in this case. So we would end up with something else, similar to our oxygen going in or water going in, and then we end up with oxygen as a byproduct. So let's take a closer look at these photosystems. When we talk about the different photosystems, there's photosystem 1 and photosystem 2. We see both photosystem 1 and photosystem 2 in plants and cyanobacteria. In other photosynthetic bacteria, we see only PS1 or photosystem 1 or PS2 or photosystem 2. So in plants and cyanobacteria, we see photosystem 1 and photosystem 2. And then in other photosynthetic bacteria, we see only photosystem 1 or photosystem 2. And then these also would be the plants and cyanobacteria, as we've already mentioned, are oxygenic. And then our other photosynthetic bacteria are anoxygenic. So we're getting a little bit more terminology here to build on this. So we see in our plants and cyanobacteria, we have both photosystems, and they are oxygenic. So when we have both photosystems, it's actually oxygenic, which means we're using water. Water is breaking, utilizing that electron to replace the missing electron in the reaction center, and then we end up with gaseous oxygen. The other photosynthetic bacteria, then we either have PS1 or PS2, and then in this case, remember, we're utilizing something other than water. So we don't have oxygen as a byproduct. We have something else as a byproduct, just as a side note, we're going to talk about it in 8.7. But a lot of these are related to these other cycles, like the sulfur cycle or the nitrogen cycle, to where these are necessary things because we need to get elemental sulfur into the system somehow. And this is a way that we can do it with anoxygenic photosynthesis. So other bacteria that are photosynthetic, and then they don't produce oxygen, but they do produce something else that's necessary for life. And we can write this down. I didn't do it previously, but if we're talking about oxygenic photosynthesis, and this is even in your text, it's written down as our carbon dioxide plus water, because remember we're talking about oxygenic, plus our light energy is going to be changed into C6H12O6, which is glucose, plus our byproduct of oxygen, and we end up with water as well. So we are just adjusting some of the water and taking electrons there, but we still end up with water, and we also end up with oxygen and, of course, the glucose. So this is oxygenic. Our anoxygenic can be written out in this way. So we also have carbon dioxide, not as much, but then we have two molecules of this H2A. Remember, the H2A is something like hydrogen sulfide or thiosulfate or something like that, plus light energy. And then that's going to be converted into CH2O, which is some version of a carbohydrate. This is the general term, the CH2O. It is the kind of placeholder for carbohydrate. You can see it's the same format up here, CH doubled and O. This is glucose, plus two molecules of whatever A is, so sulfur, for example, plus water as a byproduct. So we can see the differences there between oxygenic and anoxygenic. So now there are two more terms that we need to talk about, and that's related to these photosystems. So let's take a closer look at the photosystems so that we know kind of what we're talking about. All right, so we are going to draw this out here. So first here we have our photosystem, and in this case this is photosystem 2. And recall that within these photosystems we have these different pigments. This is just, basically a very simple illustration. And then we have our light energy coming in from the sun here. And that light energy is going to excite all of these pigments. So it excites them. Remember, it doesn't actually remove any electrons, but it increases their energy. And then at the center here we have our reaction center. In our reaction center we do have a pigment that then does lose an electron. So that electron moves up. So we're just going to say electron. And then that electron moves to an electron transport chain. So it's going to go along some proteins here. Again, very basic illustration just for illustrative purposes. So we have that electron, it moves and it goes into the electron transport chain. So similar to any other electron transport chain that we talked about, we move the electron across, moving it from protein to protein to protein. As we do this, as we move it from protein to protein, it's releasing bursts of energy. And when it releases those bursts of energy, that's in the form of our carriers. In this case, we are making ATP. So we make ATP as this electron is moving down the electron transport chain. So this electron then moves to photosystem 2. So here we are in photosystem 2. Photosystem 2 also has these pigments in it, and these pigments actually are exposed to light also at the same time our our PS1, I already had PS2 here, our photosystem 1, I apologize, photosystem 1 is also having light energy hit it at the same time. So these photosystems are getting excited as well. That energy is increasing, and then they have the reaction center as well. So we move this electron down here. The electron that we've taken from photosystem 2 actually is going to move to photosystem 1 here's reaction center. So we get that in the reaction center of photosystem 1, and then all of this energy from the sun is going to move this electron up and remove it from the reaction center. Now it's going to also go to an electron transport chain. So it's going to go to an electron transport chain over here. We have this electron moving across the electron transport chain. As it does this, it's actually going to produce NADPH. So over here we're producing ATP, over here we're producing NADPH. So we have two electron transport chains here. So when we are taking a look at this, now we can talk about those two different types of photophosphorylation processes. So we have what's called non-cyclic photophosphorylation, and this is the pathway that's used in our photosynthetic organisms when we need both ATP and NADPH. So if the cell needs NADPH as well as ATP, then it can do what's called non-cyclic photophosphorylation. So non-cyclic photophosphorylation. So this is when the cell needs ATP and NADPH. So as you can see here, if we're doing non-cyclic phosphorylation, what we're doing is going through this whole process with the PS2 and the PS1. So we're making both the ATP here in this electron transport chain and making NADPH in this second electron transport chain. Then we have the other one called cyclic photophosphorylation. Photophosphorylation. So cyclic phosphorylation. So this is the pathway that's used in photosynthetic organisms when the cell's need for ATP outweighs its need for the NADPH. So in this case, it's saying, you know what, we don't need NADPH right now. What we really, really need is ATP. So in this case, all we're going to use is the PS1, and the electron is going to return to the PS1 after the electron transport chain. So what this means is that we have our electron, and it's going to be moved up through here, and then this electron instead of going through this electron transport chain, it's actually going to go over here, and then it's going to move through this electron transport chain and go back to the PS1 and then be lifted up again, and then go back and then go down this electron transport chain again, because this electron transport chain is producing the ATP. So in this case, cyclic phosphorylation is when the cell's need for ATP outweighs its need for NADPH. So in this case, again, it's going to continue going. So right here, this process is going to be non-cyclic. So moving from here, the PS2 to the PS1, and then moving down here, giving us NADPH and ATP. And then up here, this would be the cyclic photophosphorylation. So the electron is going to be moving in a circle or a cyclic style, going back to PS1 and PS1 and PS1 over and over again, so that it can just continuously make ATP rather than making NADPH as well. So that is our photosystem setup, the PS1 and the PS2. And so recall that some organisms here right? Like plants and cyanobacteria here, plants and cyanobacteria are utilizing PS1 and PS2, whereas other photosynthetic bacteria only are using PS1 or PS2. And then also that's anoxygenic, so we're not using that H2O to get the electron from H2O and then ending up with the O2. Instead, we're ending up with something else. But either way, we require light energy. So getting back to the beginning of the chapter with kind of the general terms of, or the general process, the two stages of photosynthesis, let's kind of piece together our light independent reactions with the light dependent reactions. So what we've been looking at thus far are the light dependent reactions, right? So if we back up here, this is obviously light dependent. We have light coming in, hitting those pigments, exciting them, moving the electrons, making the ATP and the NADPH. If we remember back to the beginning of photosynthesis here, we said two stages, that the light dependent reactions is where we have light absorbed by pigments in the membranes and then it's converted into stored chemical energy. So now we know that this stored chemical energy is our ATP and NADPH. Right, so this is our stored chemical energy. So now let's take a closer look at our light independent reactions here, where we're going to utilize this ATP and NADPH, and then we're going to utilize carbon dioxide as well to make our organic molecules. So our light independent reactions. So our light independent reactions, if we write them out generally, our light reactions provided us with ATP and NADPH. And then these are short-lived, remember, stored energy but short-lived. And then we're going to be converting this into carbohydrates, which would be more of a long-term energy storage. This is short-lived energy, long-term energy. We are going to be getting the carbon for our carbohydrates from carbon dioxide. And then in this case, the waste product is going to be gas. So we'll take a look at that. So the cycle is called the Calvin-Benson cycle. So Calvin-Benson cycle by the people that figured this out. So the Calvin-Benson cycle, this is the biochemical pathway that's used for the fixation of carbon dioxide. It's located in the cytoplasm for bacteria, or the stroma in the eukaryotic organisms. So remember, when we're talking about the chloroplast, we had that cytoplasm area outside of the grana, and that was called the stroma. Remember, I mentioned that that's going to be important, and that's because this is where our Calvin-Benson cycle occurs in eukaryotic photosynthetic organisms. And then in our prokaryotic photosynthetic organisms, this is going to occur in the cytoplasm, because remember, we don't have the organelles, we just had that infolding of the membrane. So this is for fixation of carbon, and we're going to use carbon dioxide here. And then again, it's located in the cytoplasm when we're talking about bacteria. And it's located in the stroma, which is cytoplasm, but in a specific area in eukaryotic organisms. So our Calvin-Benson cycle is in three parts, or three stages. So stage number 1 is our fixation stage. And in fixation, this is where we have rubisco. So rubisco is short for ribulose bisphosphate carboxylase. And when we write out rubisco, we capitalize RUBISCO. So rubisco is ribulose bisphosphate, and then this is our carboxylase, or CO2. So rubisco. So rubisco is the enzyme that is going to catalyze the addition of carbon dioxide to our starting molecule, which is ribulose bisphosphate, or RUBP. So we have Rubisco is going to add carbon dioxide to RUBP. And remember that RUBP stands for ribulose bisphosphate. So ribulose bisphosphate is RUBP, and the enzyme ribulose bisphosphate carboxylase, Rubisco, is going to take carbon dioxide from the environment, and then it's going to catalyze the addition of that carbon dioxide to this RUBP. When it does this, it results in 3-PGA. So 3-PGA is actually 3-phosphoglycerate. So 3-phosphoglycerate is what's going to result from Rubisco taking carbon dioxide and adding it to RUBP. And that is the fixation stage of the Calvin-Benson cycle. So then we go to the second stage, which is reduction. And in reduction, we're going to take molecules of ATP and NADPH, so remember from those light-dependent reactions, and we're going to convert this 3-PGA into G3P, or glyceraldehyde 3-phosphate. And remember that glyceraldehyde 3-phosphate is an intermediate molecule in building glucose, so in glycolysis. So we actually take 6 molecules of ATP and NADPH, and then we convert this 3-PGA into G3P. Glyceraldehyde, glycerol, phosphate, glyceraldehyde 3-phosphate, G3P. So we take our 6 molecules of ATP and NADPH and we convert the 3-PGA molecule into G3P. And then this G3P can go into glycolysis. So that can go into glycolysis. But then we want to go through the third step, which is regeneration. Because this is the Calvin-Benson cycle. A cycle means that there's not a start and a stop. It's just a continuous cycle. So we take off some molecules of G3P here and we put them over into glycolysis so that we can make that carbon molecule. Remember, the end goal here is to make those long-term storage molecules. So their long-term storage molecule here that they're making is glucose. So then, however, we want to make sure that we keep the system going, so we have to regenerate it. So by regenerating, we're taking the remaining G3P that's not being put into glycolysis to make glucose, and it's going to regenerate RUBP. So it's going to be shifted and changed into RUBP, which is the ribulose bisphosphate that we saw in the first step, and then that ribulose bisphosphate is going to interact with the carbon dioxide via Rubisco to then make the next 3-PGA molecules. And then it will continue to go to reduction and then regeneration again. So G3P, the one that's not being put into glucose, is going to regenerate RUBP. And so then that's going to allow us to go again back into fixation. So if we kind of draw this out, I find that it's easier to draw out the Calvin-Benson cycle here. So we have our enzyme, Rubisco, here. Remember, this enzyme is going to take RUBP, so the molecule RUBP, and it's going to combine that with carbon dioxide. And it actually takes 3 molecules of carbon dioxide for the 3 molecules of RUBP, is what we have here. So Rubisco is going to take this RUBP and the carbon dioxide, and then this is step number 1, the carbon fixation. And then what comes of that is our 6 molecules of 3-PGA. So we have 6 molecules of 3-PGA. Then we're going to do step number 2. So remember, step number 2 is reduction. So the reduction of this 3-PGA. And do we remember what it requires? 6 ATP and 6 NADPH. So ADP and ADP plus. Whoops, 6 of them. All right, so we use our 6 ATP and we use our 6 NADPH, and we are reducing 3-PGA. When we reduce that 3-PGA, what we're going to get are 6 molecules of our G3P. So our 6 molecules of G3P. Now remember, we are going to remove some G3P, and in fact we only remove one G3P, and that's going to go into glycolysis. Then the remaining molecules, 5 G3P, are going to be shifted and regenerate into our 3 RUBP. And this actually takes more ATP here. So 3 ATP going to 3 ADP. So we just get rid of one G3P. One G3P goes to glycolysis to make glucose. The other 5 G3P are going to make 3 RUBP, and then we are now back in the cycle here. So I missed over here, regeneration is step 3. So we have fixation over here at the top, then we have reduction, and then we have regeneration. And you can see how it's a cycle because we are continuously adding this carbon dioxide, we're regenerating the RUBP, we're creating these other intermediates, and then we are making glucose, our long-term or longer-term energy storage molecule. All right, so that summarizes the Calvin-Benson cycle. And remember that this Calvin cycle, or the Calvin-Benson cycle, is used by plants. It's also used by our photoautotrophic bacteria. So remember, photo meaning energy from the sun. So of course we have our light dependent reactions where we get the energy from the sun that makes ATP and NADPH. And then autotrophic, remember that it's creating its own energy source. So it's taking an inorganic carbon source like carbon dioxide, and it's going to convert it into complex organic carbon sources, which is the glucose. So in plants, in photoautotrophic bacteria, but then also we have rubisco in our chemoautotrophs as well, some of them, chemoautotrophs. All right, so that wraps up our section on photosynthesis. You should be familiar with all the different steps of photosynthesis, the details of photosynthesis, and then where we see this happening in the different types of bacteria, like we just mentioned, and whether it's using, photosystem 2 or 1 or both, the different terms associated with that, and all of those things we just went over.